Hello everyone and welcome to MDL 130, Basic Medical Microbiology. My name is Stephanie Thompson and I will be giving your first lecture. Objectives. So at the end of this presentation and after you have completed the reading, you should be able to define classification, identification, species, genus, and binomial nomenclature, properly use binomial nomenclature in the identification of microorganisms, including syntax, capitalization, and punctuation, identify an organism's characteristics as either phenotypic or genotypic, describe how classification, naming, and identification of organisms play a role in diagnostic microbiology, list, classify, and contrast the main categories of microorganisms, and contrast eukaryotic and prokaryotic cells. So I always like to start off with the definitions, terms and definitions, and again I think it just kind of gives us a good starting point when it comes to reading. So the first one we have is taxonomy, and basically that just means that it's the science of classifying according to an organizational system. Classification is the action or process of classifying something according to shared qualities or characteristics, and to me that basically just means organizing the system by categories. And if you guys remember maybe from high school or maybe middle school, whatever, there was a mnemonic for the different categories, which what I remember as Dear King Philip came over for grape soda. So that just kind of, is your domain, kingdom, phylum, class, order, family, genus, and species. For this class, we don't really care about those top ones. We are going to just basically talk about your genus and species. So your genus is the category that has similar genetic and phenotypic characteristics. And then your species is then your most basic taxonomic unit. And same thing, this is also a collection of strains that share physiologic and genetic features. Binomial nomenclature is the naming system that we use that represents the genus and species. And then identification is the process by which we identify an organism based on its key features into the appropriate genus and species. Binomial nomenclature. So the first letter of the genus is always capitalized, and this is usually, binomial nomenclature normally applies to when you have something in writing. So the first time the organism is named, you have to spell out the whole word, or the genus has to be spelled out. After that, you can just abbreviate it to its first letter. The species name is always lowercase and is never abbreviated. However, for internal purposes, you will see that we do abbreviate it, but that's just for internal use. When it comes to actually reporting things out, we do use the complete name. And then both components are then either italicized or underlined. So as you can see from the example here, Streptococcus pneumoniae, you have your capital letter spelled out. It's in italics, and then if you were going to abbreviate it, you have to also make sure that it's either underlined or italicized. Identification. So when we talk about identification, we're talking really about microbial identification. And again, we are looking for key features that makes this organism what it is. So if something looks like Staph aureus, smells like Staph aureus, it probably is Staph aureus. However, we do have to have biochemical testing that says, okay, we are 99.99% sure that this is Staph aureus. The phenotypic characteristics are pretty easy when it comes to Staph. You have the Gram stain, which is gram-positive cocci and clusters. So these are things that you can see. Your phenotypic characteristics are your observable characteristics. And then you also have genotypic characteristics, which basically is the organism's genetic makeup. So why is this important? Well, we use the phenotypic and genotypic features to provide clinically relevant information in a timely manner. Just based off a gram stain alone, we can tell the physician that he probably needs to treat his, patient for staph. Or if it's not staph, and let's say we are looking at a gram-negative organism, then the clinician's going to want to treat the patient with a different type of antibiotic. Unfortunately, identification is not always straightforward. There are certain factors that can affect what an organism might look like phenotypically. However, usually biochemically, the organism, will fit into what it's supposed to fit into. There are times where we can't necessarily say, okay, I'm 99.99% sure this is what it is. And that's when we just kind of let the doctor know it's probably this, but it could be something else, and we're going to have to send it out for even further testing that we might not be capable of doing in the lab. Another thing that's important to know is that the clinical microbiology department is sometimes a sentinel for emerging causes of infectious diseases. And basically what that means is that we are the front line. If you think about the anthrax issue that we had, they didn't really know what it was until the microbiology department said, we have Bacillus anthracis. So we are really the front line, and we're the ones really exposed to the organisms, and that makes it really, really important when it comes to microbiology. So here we go to the main categories of microorganisms. This book is a very, very basic book. You might actually know more than what this book has to offer at certain times, which would be great. However, our goal for this course is to be able to prepare you for diving deeper into this subject when we get to MDL 251. So knowing the basics will really, really be helpful when it comes to having to know the more in-depth stuff later. So viruses. Your viruses are your most simple microbes. The important pieces here are your nucleic acid, proteins, and lipid envelope. Some of them have one, some of them don't. So then your secondary classification is whether you have DNA or RNA, single-stranded or double-stranded, whether the organism has an outer envelope or no envelope, and then also looking at the shape and size. And the other thing about viruses is that it's dependent on host cell for survival and replication. So a virus isn't going to survive on its own for very long. Bacteria are prokaryotes. So prokaryotes are simple unicellular organisms with no nuclear membrane, mitochondria, Golgi bodies, or endoplasmic reticulum. And they produce asexually. And the primary classification that we look for are the gram stain that I mentioned earlier. So you have something that's either positive or negative. A good way to remember it is that positive is purple because it has a P. Negative has the letter N, and so does the word pink. So N negative is pink and positive is purple. The other stain that we use is an acid-fast stain. And these organisms normally have a very lipid-rich cell wall. Some of the secondary things that we look at are shape, growth requirement, whether it likes to grow aerobically or anaerobically, or if it's an obligate intracellular organism, meaning that it needs to be inside of a cell to survive. And then the other thing is spore formation, and usually those are your gram-positive rods that like to do that. Fungi, or fungi, however you want to pronounce it, are eukaryotes. So eukaryotes as in you, like me and you. These are organisms with a well-defined nucleus, mitochondria, Golgi bodies, and endoplasmic reticulum. And how we classify them is either single-celled or multicelled. So your single-celled organisms are your yeasts, and your mold is multicelled. And then they all can also exist in both forms, which they call dimorphic. Then we also look at the hyphae, whether it's pigmented or non-pigmented. We look at the arrangement of the conidia, and then we look to see whether it's septate or non-septate. Parasites are eukaryotic as well. Again, we have single-celled and multicelled. So your single-celled organisms are your protozoa, which includes your amoeba, your flagellates, and your sporozoa. Your worms, or what we call helminths, can be your roundworms, which they call nematodes, flatworms are trematodes, and tapeworms are cestodes. And then we also have your bugs, which are your vectors, so like your mosquitoes, ticks, flies, lice, mites, and fleas. I know I kind of read that backwards, but same difference. The book has a really good table that kind of breaks it down to primary and secondary classification. And I think that table is pretty much sums up what you need to know about this chapter. What else you need to know? Good bacteria versus bad bacteria, or microbes, I should say. So did you know that we actually have more bacteria than cells on our body, or in ourselves, on ourselves? And all those bacteria that you find on us is what we call normal flora, and what they call in the book as your microbiome. It's very important for immune system development because as babies, we actually are born without an immune system. And so as we become exposed to things, that's how our body develops its immune system. And then later on, it becomes important in metabolic functions. So normal flora doesn't really do us any harm. If anything, it does us, does more good than harm. However, they can be harmful if they are introduced into normally sterile sites. So, for instance, you cut yourself and you get a staph infection because you have staph on you, and maybe your immune system wasn't doing so hot, so you ended up with an infection. And so what we call that is an endogenous infection. Exogenous organisms are the ones that we really need to worry about. However, there are only some that are pathogenic. And the other thing about it, too, is that it depends on the virulence factor of the organism, which is basically the necessary properties to cause disease. And I think this is all I have for today. Yep, that's it. I will see you in the next lecture.